Hi everyone, welcome to Bengaluru. I am Anik Dhanaraj, a science fellow in Bayer, and I'm currently working in the Skippy team, that is Science, Competitor and IP Intelligence, and I will be your guide for this video. So, gear up for an exciting journey as I take you for a private tour of our genotyping lab. But before we get there, I would like to introduce you to Dr. Bindu Nair, who leads the genotyping lab.
Hi, it's wonderful to meet you all. We are always excited to meet new people who are interested in the science that we do. This lab has people with diverse expertise like plant physiologists, molecular biologists, biotechnologists, automation engineers, and many more. All of us are striving towards helping breeders speed up their plant breeding programs in India by generating molecular marker data. Let me now hand this over to Anik, who will guide you through the Genotyping Lab, the place where magic happens.
Hey, so together we will witness some of the activities that help breeders speed up the development of vegetable, corn and rice hybrids in India. At the Bayer Crop Science Division, our passion is for innovation. Our motto is not only to grow enough, but to grow better for our planet and its people. With leading research capabilities in plant breeding, biotechnology, crop protection, and data science, we deliver tailored solutions to farmers faster than ever before. We leverage the power of technology for agricultural sustainability that can increase productivity while reducing environmental and social costs. One such pioneering technology that we use is plant genotyping. Let us first see the activities that our scientists perform in the genotyping lab. To begin with, we collect the samples, followed by lyophilization and then powdering. The next step involves DNA extraction and then polymerase chain reaction or PCR setup. After this, we perform the PCR and read the PCR signal in the array. Once this is done, we finally score the plots to check if we got the desired results. Let me now take you through an actual genotyping process. The samples that are analyzed in the genotyping lab are leaf bits, which come in as fresh tissues in blocks. Each block can hold up to 96 samples, which will be analyzed together. The fresh samples are first freeze-dried by placing them in a lyophilizer overnight. After they are dried, beads are added and shaken vigorously to powder them in preparation for DNA extraction. For DNA extraction, known quantity of extraction buffer is added to each block using the 96 well head in a liquid handling system. The tips are first loaded onto the 96 well head, which then moves to a trough containing the buffer. The buffer is first aspirated and then dispensed into the blocks that have the powdered leaf samples. This step can be repeated many times to transfer buffer to other blocks. This way, many samples can be analyzed at one time in a high throughput manner. The liquid handler can be programmed to add buffer to numerous blocks at one time. Once the buffer is added to the blocks, they are capped and homogenized by placing them in a homogenizer, and then they are ready for spinning in a centrifuge. The plant debris are separated from the extract by spinning the blocks in a centrifuge. During centrifugation, because of the centrifugal force, the plant debris collect at the bottom of the wells in the blocks. Equal number of blocks are spun together to ensure that they are balanced during the spin. After centrifugation, the supernatants are separated from the pellets using a liquid handler, and the DNA is precipitated in these blocks. The DNA pellets in the blocks are then resuspended in the buffer using a dispenser. The dispenser will add required amounts of buffer to all the wells in the block to get them ready for the PCR setup. In the dispenser, the blocks are first placed on a tower, then they are automatically lowered to a conveyor belt, which moves it to a dispensing unit where the required amount of buffer is added to each of the wells in the block. The DNA pellets dissolve in the buffer and then they are quantified and used for PCR setup. The PCR reaction is set up in nanoliter volumes on PCR tape. DNA is first transferred from these blocks onto the tape, after which the PCR mixture is dispensed into the wells. The Nexer is a highly sophisticated liquid handler that can add nanoliter volumes of reagents for the PCR with great accuracy and speed. Once the PCR mixture is added to the tape that has the DNA, the tape is then sealed and spooled and made ready for the PCR machine. PCR is carried out in a hot water bath with the required temperatures for the PCR reaction to occur. A robotic arm that holds a spool of tape that has the DNA and the PCR reagents are immersed in a water bath with the required temperature. It is here that the DNA is first denatured, then the primers anneal to the DNA strands and they start extending. The entire PCR cycle is repeated many times to complete the PCR reaction. By carrying out the PCR reaction in a hot water bath, we can set up PCRs for a large number of samples at one time. Once the PCR is complete, the tape is read in an arrayer. The arrayer reads the presence of the PCR products in the tape and the results appear on a plot. The plots are then scored and the results let us know if the SNP markers that we are interested in are present in the plants that are being tested or not.
So, in a way, genotyping helps breeders develop superior hybrids quickly when compared to traditional breeding by enabling them to identify plants with superior traits or characters that are linked to the molecular markers. Furthermore, it allows us to conduct high-throughput genotyping analysis in a timely and cost-efficient manner.
For us, genotyping is not just about development of superior hybrids, but also transforming agriculture and changing lives of millions of farmers and bringing a smile on their faces.
Using insights from genotyping, we enable plant breeders to develop various hybrids with different traits for the Indian farmers,
thereby abiding by our scientific mission of innovation for the benefit of society.